you would make a little bit more of the ATP. So this is showing what cyclic flow looks like. Cyclic flow, and this is kind of showing energy level, is basically when this only shows photosystem 1. But the photosystem 1, when it passes its electrons to ferredoxin, ferredoxin does not drop them off at NADP+. Instead, it's a mobile unit, so it can move and drop them off with plastoquinone and then continuously go through this cycle, which is creating that gradient, which allows you to make more ATP. So that would be cyclic flow. Linear flow looks like this, where this actually shows the whole thing from the very beginning with photosystem 2, the excitation, and then as they move down, you see that they're losing energy. They get re-excited, handed off to ferredoxin, and then it lets them go at NADP+. And that would, you can see how that's linear. It drops them off. Okay, and this emphasizes the final acceptor for the electrons in photosynthesis is what? What takes the electrons at the very end? NADP+. How about cell respiration? What was the final acceptor for cell respiration?
Oxygen.
Oxygen. Okay. Does anyone have any questions? I know we went over this at the end of like 2.5 hours of lecture yesterday. So any questions on this light reaction part? The light dependent reaction. Okay. So again, I'm going to try to put us back into the context of in the chloroplast, where are we at, and then what are we making? Oh, sorry.
Excuse, how often does it switch between the cyclic and linear flow?
That's a great question. There has to be factors that are sensing ATP levels and triggering to switch. I don't know all the molecular details of that, but there's got to be some kind of a mechanism that is sensing ATP. And this will come into play with Calvin cycle, because let me show you the Calvin cycle briefly. With Calvin cycle, you need more ATP. There's 9 ATP and 6 NADPH for every 3 carbon dioxides. And so because of that slightly more ATP is needed, it's got to kind of sense that, and then it has to switch between the two. That's a great question. Other questions that people have about the light reactions? Okay, so kind of putting us back into the chloroplast and where we were talking about all of this. Can anybody tell me where these proteins are found within the chloroplast? Thylakoid membrane. Yeah, so they're right here. If we were to blow this picture up, that's what that would look like. So the light reactions are all taking place within the thylakoid membrane. If you had to label on here which region of the chloroplast would have a low pH, where would you label as being, remember, low pH means high proton concentration. Which part would be building up? Yeah, the lumen. So we would say here, this is low pH, meaning that we have a whole bunch of protons. What about for if I were to draw over here, mitochondrion, where would we label low pH in a mitochondria? Where the proton gradient is building up.
Inner membrane space.
Yes. So this space in here, the IMS, this would have the low pH. This would have all of the protons. So just to kind of like put these two in context, here's where the electron transport chain proteins are. In the mitochondrion, we would see the electron transport chain complexes. They'd all be in the inner membrane. So we're kind of like drawing the connections between these two. All right. So the products for the light reaction, remember we said there's some of the major goals for this light reaction. One thing that we're trying to do is collect light. So this is, we're absorbing the light energy in this step. What are other products that we're trying to make through the light reactions?
NADPH.
Yeah, so we need to produce NADPH and what else?
ATP.
Yeah, ATP. These two are needed for the Calvin cycle or for the carbon reactions. So you have to have the light reactions in order to be able to produce the sugars that we're gonna get out of the Calvin cycle. And then finally, the other thing that is happening in the light reactions is that we are splitting water and we're producing oxygen. So we're producing oxygen gas at this stage as well. Okay. So now we will compare this. I have a worksheet that is a good comparison. I'll come back to that. Let's just talk about the Calvin cycle for a second. I can do a big overview of this and you don't need to know the details of the steps. Our textbook is actually pretty vague about the Calvin cycle as well, but it has it broken into 3 basic steps. The first step is fixation, and that is where you're basically taking CO2 from the atmosphere and you're combining it with another carbon compound. That's why it's called fixing. It's taking a carbon and fixing it with another carbon compound. The carbon compound that you fix with is called ribulose bisphosphate, and there is an enzyme that does that is called Rubisco. So this is what goes in the blank. Rubisco is probably the most abundant enzyme on Earth because of the biomass of all of the plant material that we have. So anyway, we have a ton of this enzyme. It takes a one carbon CO2 plus 5 carbon RuBP. As you can see, we're going to make a 6 carbon compound when we do that. And that is going to be very unstable. So let me just show you the cycle briefly. For fixation, we take these red balls are carbons. So we have 5 of these combined with a single carbon here. That's going to give us 6. And because we look at this for turning this cycle 3 times, we have to think about, in your mind you can think about an array where you have like 3 of these. Okay, and each of these combines with a carbon from carbon dioxide. So now you have 3 6-carbon compounds. That's extremely unstable, and so it immediately breaks. These break into 6. You can see here 6 3-carbons. And so that's where you get this right here. 6 of these 3-carbon phosphoglyceraldehydes. Don't need to know the names of all these. PGA is fine. The next step is called reduction. So reduction, we're adding electrons. So what is our reducing power? Where is that going to come from? Well, NADPH is our source of electrons. So we add phosphate groups and we add the electrons to reduce this to 6 molecules of G3P. Now, does G3P sound familiar to anybody? Right, that was found in glycolysis. So G3P is that important 3-carbon sugar that it's found both in glycolysis and it's also found in photosynthesis. Now we have 6 of them here. 1 of them gets to be shuttled off to be made into a sugar. But the other 5 have to be recycled to get our RuBP starting material again. So the last phase is going to take these 5 G3Ps, which if we have 5 3-carbons, we've got 15 carbons. They get rearranged. We have to add in some phosphates. We get back to 3 5-carbons. Does everybody see how the carbons were conserved there? And 1 of these guys gets to be shuttled off to make a sugar. How many times would this cycle have to turn to make 6-carbon sugar? If each time it turns 3 times, we get 1 3-carbon sugar. Anybody see? Somebody said it 6 times. So the cycle itself has to turn 6 times to make 1 glucose. This is an extremely inefficient cycle, but somehow in evolutionary history, the decision was made to go this route, and then as we know now, plants use Rubisco to do this process itself. Even though it's very inefficient, it is the way that it is done. So you turn this 6 times just to make one glucose. Okay. So going back to this, just make sure we covered all of the blanks here. So we talked about reduction stage where we have 6 molecules of 3-PGA. They get phosphorylated and reduced, yielding 6 one of them gets to be shuttled off to be made into sugar. The other 5 will be recycled to regenerate ruby. That's still basically the level that, in this class that we're gonna have to know this carbon cycle. The other thing to note is, where is this taking place in this for our class? Where are all those enzymes that we would have here, like Rubisco? Anybody remember? Which part of the chloroplast? It's in the stroma, yeah, I think somebody said that. So in the stroma, this is where the carbon reactions would be taking place. And actually it makes sense, because if we go back and look at this light reaction, the NADP plus reductase enzyme is found in the stroma. The NADPH would then be directly produced in the stroma. The way that this ATP synthase is oriented, you can see that the proton flow is going to go this way. The ATP is produced in the stroma. So everything that you need is right there. You have NADPH, you have ATP, you get CO2 that just diffuses in. Okay, so that is sort of the summary for Calvin cycle. Does anybody have any questions at least about the Calvin cycle itself? No? Okay, this is a really good review, I think, for making sure you have clear respiration and photosynthesis. So we're gonna do this. This will help review for the test. So I wouldn't ask you to do it if I didn't think it would help. And at the bottom, it asks you to draw chloroplast and draw mitochondria and label where all of the processes take place. And I'm telling you right now, I always put that as one of the short answer questions, so it's good to practice. So why don't you work on this and check your answers with people next to you, and then we'll go over this worksheet. And then the time that we have left, I do have some questions that we can also go over.